Good morning, everyone. Shout out to Tony. He mentioned the FOXP2 gene the other day, and I'm only gonna tell the truth. So let's get into it. This study was done that challenges evolution of FOXP2 as human-specific language gene. So he mentioned FOXP2, and I didn't know what FOXP2 was. I'd never heard of it. I knew of another gene related within the same forkhead protein family known as FOXA2. I may do a video about that. I'm not sure yet. But they were speaking on the development of the human species. And he made absolutely, as we know, I'm talking about bratwurst. He made no sense because the bratwurst elf was talking about Neanderthals going extinct 3 times, which we know is not possible. So I'm not only here to debunk his claims because that would take no more than 30 seconds, but I'm here to reiterate the fact that we have millennials out here who are reading and who are studying, and I'm very proud of them for going above and beyond to look into what the hell is going on and able to bring sources when necessary. So that's important. So the analysis of their genetic data from a diverse sample of modern people and Neanderthal, saw no evidence for recent human-specific selection of FOXP2 and revises the history of how we think humans acquired language. So they're saying that although FOXP2 has been implicated in affecting speech and language, it may not be as critical to language as what we recently thought. And that's fine. There's no gene that's set in stone, right? But we also realize that in Cell, they have the data for this, which I'll show you in a minute. It was published last year, and so they show here. Here's one instance that's supporting their claim. FOXP2 is highly expressed during brain development and regulates some muscle movements, aiding in language production. When the gene isn't expressed, meaning it's knocked out, it causes a condition called specific language impairment, in which people may perform normally on cognitive tests but cannot produce spoken language. So these are people that you would consider mute. So and they also have the physiology there. The anatomy is intact, but they're not able to produce words, produce sounds. FOXP2 has also been shown to regulate language-like behaviors in mice and songbirds. So mice usually just squeak. Sometimes they make a squealing sound if they're in pain. It's a stress signal to the other mice. The babies do this even when they're small pups, just a few days out of the mother. They'll start squealing and squeaking. Songbirds do this, of course. Most birds do this as a form of communication. So we know it's there in other species other than humans. They come down and talk about in the past 5 years, several archaic hominin genomes have been sequenced, and FOXP2 was among the first genes examined because it was so important and supposedly human specific. But what they're finding, and this is what Tony was bringing up, what they're finding is FOXP2 has also been linked with Neanderthal, which is a human-like, a humanoid species. And current people of the white Caucasoid race, for instance, do have some percentage of Neanderthal DNA. I'll leave that up to you to look up on your own. Because if you come to my channel, you are going to have to read. There's a lot of information that may seem boring. But one thing I do know is that if you can't sit for 30 minutes to an hour a day reading, then it's very difficult to engage in certain conversations where policy comes into place, where economics come into place, because we have to get ourselves in the mode of reading things that may not interest us at first. It all takes practice, and the mother of all learning is repetition. Repeating that practice of learning new things and starting to merge ideas together, right? So you can come up with solutions to problems. Coming up with solutions to problems is something that is time consuming, but necessary. Okay. All it boils down is to having discipline. So this is the actual study where they say there's no evidence for recent selection at FOXP2 among diverse human populations. Now, I'm going to show all the authors here, and I don't have their support here listed, which is a red flag to me when they don't openly show their support, openly show who gave them the money to do the research. But the highlights say that no support for positive selection at FOXP2 in large genomic data sets. The sample composition and genomic scale significantly affect selection scans. That just means that the samples that they got affected how they were able to analyze the sample. So genomic scale is you're looking at the entire genome, every chromosome, and you're comparing genes within the forkhead family, FOXP2, FOXA2, FOXM1, and so forth. You're comparing them all to that particular gene. Then they say an intronic ROI within FOXP2 is expressed in human brain cells and cortical tissues. I forget what ROI means. I think it's region of interest. But the introns are in between exons, and introns are able to control how exons function. So the exon is the functional part of the gene. Intron is an internal control. And an intron is necessary because it is a point where after the exon stops, you have an intron. An intron is necessary between genes because that is also where the proteins involved in DNA replication and RNA production of RNA, they stop. They have to be replenished. This is not like something where they just keep going on and on. They have to be replenished. Different proteins have to come in to aid in the production of new exons, the next exon and the next exon and so forth. This ROI, they go on to say, contains a large amount of constrained human-specific polymorphisms. Polymorphism is nothing but a change or a mutation in the gene, and poly means many, so many changes within the gene. That's all that means. Don't let these big words, throw you off. Just break it down. Break it down. Just break it down. I'm gonna go down here. They talk about the graphical abstract. Another thing I don't like about this paper is and that's my fault for choosing it, but it was one of the first ones up there, and the date really got me because I was like, wow, this is published August 2nd, 2018. This is very recent. They're still studying this concept of FOXP2. Their graphical abstract talks about a lot of things they did, but they don't show you a lot of their data. They give you a graphical abstract, and that's really nothing. Then they tell you down here, you need to purchase one-time access or subscribe to sell. Now, at most people's jobs, you don't have to do that. You just, get the access. I don't have access. So I typed in the article title here in NCBI. NCBI is my go-to for a lot of the research I look up and review. And it's also, oops, sorry for that. It's also something that I use to design my primers for PCR. So they say here, there's no evidence. So I went to Cell Press, and that's this next link here, and I get the same thing. So I'm like, what the hell? They do not want me to know what they have to offer. So one way you can get around this is to contact them directly, and they will send you scanned images of their data, or you go do what I do, which is the back door way, is to go on Google, type in the title of their article, and type images and go to the images directly. So you see the same image here that they have on the website. But you also have other images. For instance, the first page of the article is here, because a lot of times these articles have to be posted on Twitter. Then we get into some of the figures that they have. So this is a way to go around, and I've spoken about this on my channel before. It's the way to get around them not wanting to put their data out there, and they don't have to. You know, if they want to charge you for their data, they're gonna charge you. You know, that's their prerogative to do that. Especially since they are doing research at this seems like a very high level. This looks like mass spec again that they're doing. I'm not going to go through every single figure because I would expect people to, if you're interested in this type of research, to go to it and to see the way I'm looking. This is how scientists look things up. When they can't find the data, they go in the back route and look it up with Google Images. Okay. So they talk about here in this figure, for instance, FOXP2 targets show evidence of probably positive selection in FOXP2. Okay. So you have CEU. I'm not sure what that means, but they compare FOXP2 to the mouse brain, the human brain, and the human neuronal cell. And you find that you have similar expression of FOXP2 in the mouse brain and the human brain. I'm not sure what the other abbreviations are. I have to look that up. But it does indicate to me that FOXP2 is conserved within humans and mice. It may not be conserved in those who express Neanderthal DNA. So they are showing that there is no connection between FOXP2, and it is a connection between FOXP2 and language, but not FOXP2 as far as Neanderthal. So people with Neanderthal DNA may be quite disappointed in what they're showing here. And there's a figure here. They go into detail about FOXP2 variants are not human specific. They're trying to determine the Denisovans and Neanderthals, what happened to change the anatomy of modern humans, especially the skulls and therefore the brain. They see that over time, there doesn't seem to be evidence for recent selection in FOXP2 across diverse populations. So they need to develop a new hypothesis for how language developed. And the way they determine this was they see that there's an intronic region in FOXP2 that's enriched for human specific single nucleotide polymorphisms. Human, not Neanderthal, bratwurst, but human specific single nucleotide polymorphisms, also known as SNPs. And this is where you have your genetic diversity. They're showing this different colors, people of different colors. I don't know anybody that's purple, but the colors are representing different genetic diversity as well as it also relates with melanin as well, because FOXP2 is, like I said, within the family of FORKid. A lot of these genes are linked with melanin. A lot of these genes are also linked with metabolism, neuroendocrine differentiation, the differentiation of muscles, the differentiation of your nerve cells, the nerve system. So all these genes are pretty critical to your survival and development. Anyway, I don't want to go on too long. I have these links in the description box. Check them out. I'll holler at y'all later. Once again, have a great Friday. And you might see me later on this evening on a hangout or something. But if not, be safe, and I'll talk to you soon.